Welcome everyone to a new video. So this is my second video on the antibacterial agent, and in my first video on antibacterial agent I talked about the sulfur drugs. I talk about the mechanisms by which various mechanisms in fact by which the antibacterial agents act. So in this particular video I'm going to specifically talk about the penicillins and its mechanism of actions and the application of penicillins. But what are the applications? So all the content that I'm going to discuss in this particular video has been taken from the book An Introduction to Medicinal Chemistry by Graham Patrick. So if you're someone who's really interested in learning and understanding more about the medicinal chemistry, then I highly recommend this particular book. Okay, so now let us start the video to see what are we going to talk about in this particular video. What are the content of the video? So in this video we are going to talk about first of all the history of the penicillin, and then we are going to look at the mechanism by which the penicillin act, and then what about the resistance to the penicillin, and then the methods by which various penicillins and analogues can be synthesized, and the structure activity relationship, means what are the important moieties, what are the important structural features of penicillin that are absolutely important for it to act the way it does. And then we are going to look at the various analogues of penicillin and acid resistant penicillin and beta-lactamase-resistant penicillin. And then we are also going to talk about the broad spectrum penicillin, more specifically about aminopenicillins, carboxypenicillins and ureidopenicillins. So let's start the video and first of all discuss the brief history of penicillin. So if we go back into the history, the entire story started in 1977 when the Pasteur and Joubert, they discovered actually that certain molds, they produce toxic substances, and these toxic substances, they kill bacteria. But what they found that these substances were too toxic for human and they were of no clinical use, but it gave some directions that you can have certain substances or certain molds that can have potential application in the future. And now flash jump to 1928. What Fleming, when he kept one of his bacterial culture left open for days, found that it had it was infected by a fungal colony, and he thought that that's a very important discovery. So he identified the fungus, and it showed that it is a very relatively rare species of penicillium. Okay, so he spent several years, Fleming, and investigating the novel antibacterial extract, and what he found that extract had significant antibacterial property, and it was remarkably non-toxic to mammals, means it was not toxic to humans, right? So that was so remarkable about that particular discovery. But the unfortunate part that he was not able to isolate and purify the actual ingredient which was responsible for the activity. So the problem of isolation of penicillin was actually solved in 1938, and it was done by Flory and Chain, and they used processes like freeze drying and chromatography. And then by 1941, Flory and Chain, they were able to make enough of crude extract of penicillins so that you can carry out the first clinical trials, and there were further development in process that were made, which were basically developed in the United States. And by 1945 they had enough supply of penicillin to treat the casualty arising from D-Day landing, which is nothing but Normandy war that we are talking about. However, the structure was still quite a controversy, so that was solved in 1945 by Dorothy Hodgkins, which used X-ray crystallographic analysis to give the structure. And in 1947, first synthesis of this highly strained molecule was accomplished by Sheehan. However, this synthesis was so long that it had no commercial value. Okay, so this was the brief history of the penicillin. Now let's look at the structure of the penicillin. So here what we have, this is how the basic structure of the penicillin looks like. So here what we have is a beta-lactam ring. So this is the beta-lactam ring, and this is nothing but a thiazolidine ring, and the fragment that is there present in the blue picture, it is called 6-aminopenicillinic acid. It is also called 6-APA. Now, Beckham, what he did, he was able to isolate this biosynthetic intermediate of penicillin. And how does it help us? If you can isolate this particular intermediate having this amine moiety, now you can synthesize huge variety of penicillins by simply acylation reaction. So if we carry out a disconnection here, what we have is an acyl chloride, here what is an amine, we can carry out the acylation reaction to give the desired penicillin. Similarly, if we have R group here, which is the benzyl penicillin, it will be called penicillin G. If we have R group as phenoxymethyl, then it is called the penicillin V. So depending upon the penicillin that we want, we can simply start from this particular intermediate, which is 6-APA, which is a huge discovery actually, and carrying out the acylation reaction. So later on, when we are going to discuss the structure activity relationship, we are going to talk more about the structure of the penicillin and its properties. If someone talks about the biosynthetic precursors of penicillin, how does the nature makes penicillin? Basically, nature takes advantage of two amino acids, that is cysteine and valine, and it undergoes the whole biosynthetic pathway to give the penicillin. So it is very, very important for you to understand that it is cysteine and valine which are involved in the biosynthetic pathway, or they are the biosynthetic precursors for the synthesis of penicillin. Now, to understand the mechanism of action of penicillin, I will go a little slow. It is basically, there are 3 slides in which I am going to slowly discuss about the mechanism of action of penicillin, and it is very, very important to understand. So I'm gonna go a little slower and make sure that you guys understand. It's very easy. So first of all, we have already discussed in the last lecture on the antibacterial agent that bacterial, they have cell wall. And why do they need cell wall? Because they need to survive in the harsh environments where there could be changes in the pH, temperature of the osmotic pressure. Now, this bacterial cell wall, it is made up of peptidoglycan. So as the name suggests, if we divide into half, this is peptide, this is glycan, means sugar. So the sugars that we are talking about here is nothing but is an acetylglucosamine and an acetylmuramic acid. So this is NAG, NAM. So this is how they are, you have this NAM, NAG, NAM, NAG, and they are interconnected, this sugar, and then all the amino acids actually that are connected to this sugar. Now, there are more than 30 enzymes that are involved in the entire construction of this bacterial cell wall. But you have to understand, the most important step is the last step, which is the interlinking. So this is the interlinking that takes place in the last step. Okay? So it takes place between the it is nothing but a pentaglycine linkage. Now you can clearly imagine, as soon as there is an interlocking takes place, the strength of the cell wall enhanced manifold. So it is basically this final interlinking which is inhibited by the penicillin. And once this interlinking is inhibited, then you can clearly imagine the water enters, it causes the cell to swell, and as a result of which the cell dies by cell lysis. So that is how the penicillin acts. Okay, so what are the important takeaways from this one? That the bacterial cell wall is made up of peptidoglycan layer, and there are more than 30 enzymes that are involved in the biosynthetic synthesis of this bacterial cell wall, and it is the last step which is inhibited by the penicillin, which is nothing but a cross-linking, and cross-linking is something that provides strength. Now in the next picture we are going to look at how this happens very briefly, and then we are going to look at the exact reaction that happens into the enzyme pocket. Now let's look at very general picture. So here what we have is nothing but this is the sugar backbone, this is the sugar backbone, and this is one of the chain, this is the other chain. So what happens, this enzyme which has a serine moiety, and we know serine moiety has this OH groups, and what it does, it attacks the alanine, one of the alanine is replaced. Okay, and this particular enzyme is nothing but this is transpeptidase enzyme. So transpeptidase enzyme has these serine moieties which attack. Okay. Now, as soon as you have this D-alanine inside the enzyme pocket, the other glycine. So what we talked about, pentaglycine. So this is 1, 2, 3, 4, 5. So this is a pentaglycine chain. This glycine comes, it attacks the D-glycine, and the enzyme leaves. So what this enzyme has facilitated, what this transpeptidase enzyme has done, it has helped in interlinking. Okay. So this bond formation between the glycine and D-alanine has been facilitated by the enzyme transpeptidase. As a result of which, this cross-linking has occurred. Similarly, you can see a huge, an enormous degree of cross-linking taking place, which is mediated by this particular enzyme. And it is this cross-linking which provides strength to the bacterial cell wall. And imagine what this penicillin does. Penicillin inhibits this particular transpeptidase enzyme. So if you inhibit this first step in itself, it means there will be no cross-linking that will be taking place. Now, this entire diagram now, we are going to understand now what is exactly happening inside the enzyme pocket. So here on the top one, we are going to see how the cross-linking takes place inside the transpeptidase enzyme. So this particular transpeptidase enzyme is bound to the outer surface of the cell membrane, and it has basically serine enzymes in its active site. So these, as a result of which, it is also called the serine protease enzyme. Now, what does the serine protease means? It means that it belongs to the class of enzymes which are called serine protease. So it means that it has the serine moiety in its active site, and what it does, it hydrolyzes the amide bond. So this is exactly what it does. So as soon as D-alanine and D-alanine approaches this particular enzyme active site, what it does, this serine moiety attacks to result in the formation of this particular intermediate. So the other pentaglycine moiety, it attacks, and what it does, it leads to the formation of this cross-linking, and everything in this particular step is happening inside this active site of the enzyme. Now, what happens? How does the penicillin inhibit? Penicillin adopts a very similar conformation which D-alanine and D-alanine has. For example, this particular conformation and penicillin has a very similar conformation. So the transpeptidase enzyme gets confused, as a result of which, what it does, it attacks the penicillin. It takes the penicillin inside its active site, and as soon as that happens, we know it is cyclic in nature. As a result of which, this entire big molecule stays bounded inside the active site, because what you have is a carboxylate anion, and what you have is a lysine moiety as well. As a result of which, this entire active site is being now occupied. So the glycine moiety just cannot attack it, right? It is just not available. So the entire active site of the enzymes are now blocked. So there is no cross-linking that is possible. So that is how the penicillin inhibits the action. So here, whatever I have said is written. All the information is written here. Now, one of the reasons actually which explain why the penicillin is not toxic to humans, the reason why is because it is highly selective to D-alanine and D-alanine. Okay, in our body also we have a lot of serine protease enzyme, but since penicillin is only selective towards the D-alanine and D-alanine, and in our body we do not have D-alanine, as a result of which it is not toxic towards mammal, and it only and selectively attacks the D-alanine linkage of the enzyme. Okay, so that is the reason why penicillin attacks, and that is how it basically acts as an inhibitor. Now, what about the resistance to the penicillin? Now, the overuse of the penicillin has caused a lot of bacterial resistance. Now, what the bacteria has got smarter, as a result of which there is an enzyme which bacteria produces, which is what we call as a beta-lactamase enzyme, and it has mutated from the transpeptidase. So means beta-lactamase and transpeptidase, they are very similar in nature, but it is slightly mutated. So what it does is basically this beta lactamase enzyme, before this penicillin can reach the active site of transpeptidase, what it does, it cleaves this particular carbon nitrogen bond, as a result of which it opens the penicillin before it reaches the active site, as a result of which it completely deactivates the penicillin. Okay, so it is no longer useful, and this process is done very, very efficiently by the beta lactamase enzyme. It means it can efficiently hydrolyze 1000 penicillin molecule per second. You can imagine how efficient this beta lactamase enzyme is. Okay, so what are the approach that scientists have developed? So they have developed compounds like these, which are what we called is beta lactamase inhibitor, and more specifically clavonic acid. I think you must have seen when you go to the pharmacist, and here I'm more specifically talking about the countries like India, where doctor give you let's say drugs like amoxicillin plus along with the potassium clavonate. So if you look at the tablet, if you look at the packet, you can clearly see there will be a salt of amoxicillin plus potassium clavonate. So the role of potassium clavonate or the clavonic acid is to make sure that whatever beta lactamase that is present inside your body, okay, is basically being consumed by this clavonic acid, because beta lactamase has a very, very strong affinity for this clavonic acid. So what does this clavonic acid contain? First of all, it does not have any amino-acyl chain. So there is no amino-acyl chain at position number 6. Second, what we have is a beta lactam, right? We have oxazolidine ring, right? And here the oxygen has been replaced by the sulfur, and instead of 2-methyl group, what we have is a double bond here. So these are the few changes that are present in the clavonic acid. So it's the sole role of all of the clavonic acid, or the mostly in the drug it is available as potassium clavonate. It's to make sure it inhibits beta lactamase enzymes. Okay, so these are nothing but beta lactamase inhibitors. So this is very, very important to understand. Now, what are the methods by which penicillins analogues can be synthesized? As I said that Shehan, he did the synthesis. It was just too long to have any practical use. But the great thing was the Beckham discovery of biosynthetic intermediate. So the biosynthetic intermediate that we talked about was nothing but this 6-APA. So as soon as you can isolate this 6-APA, what you have to do is basically the acylation reaction, and it can give you a huge variety of synthesis. So this is called the semi-synthetic method, where what you are just isolating this particular intermediate, okay, via various methods, and then carrying out the next step of the reaction in the laboratory, whereas the synthesis before that is usually done in the biolab, because what you are doing is basically isolating them. Now this is very, very important slide. Here what we are going to talk about the SAR studies. So these are all called the structure activity relationship studies. So here they specifically talk about what are the important features about the penicillin, and what are the absolutely essential things that has to be present in the penicillin for it to be an effective drug. Okay, so the first thing that it talks about, the strain beta lactamate ring is essential. It means that this beta lactam moiety, it has to be present. You just cannot have any other. You cannot have a 5-membered lactam. It has to be beta lactam. You cannot have gamma lactam, right? That is important. The second is free carboxylic acid is essential. So this carboxylic acid has to be free. It cannot be it cannot be like an amide bond, right? It cannot be an amide. It has to be free. Now why it is free? Basically because this is usually ionized. So usually penicillins are administered as potassium or the sodium salt, and this carboxylate anion, it interacts with the nitrogen of the lysine moiety, which we saw, because that is how it occupies the binding site. So that is the reason why this free carboxylate anion is essential. Now the next is this particular bicyclic system. Having this bicyclic system is absolutely important. Okay, what has been found that greater is the strain, there greater is the activity of the this particular molecule. Means having this beta lactamine at 5-membered ring, which makes this particular system very very strained. But it is a it is a very very fine balance, I should say, that which causes this particular molecule to behave the way it does. Okay, and then having this acyl amino side chain, it means having this particular amide bond is very very important. Means it has to be present. You cannot have an ester here, you cannot have a nitrogen, you cannot have an R here. It has to be amide, which is very very important for its activity. Usually what you will find is next is the sulfur, which is usual but it is not essential. So you can have other heteroatoms, but since sulfur is mostly present, so that is fine. The second is the cis stereochemistry of this one. So here the stereochemistry has to be cis. You can have a different substituent here at this position, can be methoxy. You will see certain drugs where you will see a methoxy here, but the stereochemistry here has to be cis. Both the hydrogen have to be cis to each other. So these are the certain structure activity relationship which a drug must have if we want to classify it as a penicillin, and it has to be a very effective drug. Then these are the feature it must have. Now what is the reason for this structure activity relationship? How do we establish structure activity relationship? Basically, over the years the scientists, they make a huge variety of thousands of compounds, and then they test them for their bacterial activity, and on the basis of that data and results, they come up with this conclusion or this gist of the information, that what are the absolute imperious features in a particular molecule. Means what are the things that we can touch, what are the things that we cannot touch. So this is what we gain from structure activity relationship. Now we are going to look at that how the penicillin analogues, they became successful. They were too important actually. There were certain issues that were involved with the penicillin. The first one was the acid sensitivity, that it is sensitive to acid, and the second was beta lactamase sensitivity, which we talked about, and also the limited breadth of activity. There was a very very limited activity it had. Now first of all, we are going to talk about the acid sensitivity of the penicillin, why it is sensitive to the presence of acids. One of the reason is the ring stain. Okay, so what happens is, basically, in under acidic condition, what happens, this particular water, it attacks this particular carbonyl. Okay, as a result of which it O negative, so water attacks, so this negative charge come back, so it opens the ring. So this has been mostly found in the case of the penicillin G, where the R group is nothing but benzyl. And under acidic condition, they found that it opens to give this particular compound. So if it just gets open, it means it's no longer an efficient drug. So the first reason is the ring stain, because these two rings are so stained that this beta lactamase, it just wants to get relieved from the stain, and it would just get open and would no longer wish to be in that strained mode. The second is highly reactive beta lactam carbonyl group. Now this particular carbonyl group in beta lactam, okay, this carbonyl group in beta lactam is a carbonyl. It is not actually a true amide, although you might say ki here what you have is a nitrogen, here you have an amide, it might undergo conjugation, but that's not the case. Okay, so usually in the tertiary amide, the lone pair of electron on nitrogen and then these pi electron, they are in the same plane, as a result of which they can undergo conjugation. Okay, so this type of conjugation possible only when the bond angle is 120. Okay, means they are in the same plane. Whereas in the beta lactam, this is the half book conformation, right? That's what I said, half book conformation it has. So it has a folding structure. So these lone pair of electrons on nitrogen, they just cannot undergo conjugation, because for this particular conjugation to take place, then this ring has to have this 90-degree angle, which is just not possible. It just cannot become flat. this type of a structure is going to be impossibly strained. That's just not possible, as a result of which the carbonyl of the beta lactam behaves like a carbonyl rather than a amide bond. Okay, that is the reason why it just gets open very quickly. Third thing that we are going to talk about is the influence of acyl side chain, which we call neighboring group participation. So when we have is an R group, which are usually electron donating, or which are like just the simple benzyl group, what it does is basically the lone pair of electron on nitrogen on this amide, they come, this oxygen attacks. So these are the kind of rearranged compound like penylic acid or penicillinic acid. So these are the various type of fragmented compound you may get, and usually this happens in the case of the penicillin G. So depending upon the kind of acyl side chain you have, these are the side products that you may get. Okay, this as a result of the neighboring group participation. So the three problems as acid sensitivity is due to three problems. The one is ring strain. The second thing that you should know, it is because the beta lactam carbonyl behaves like a carbonyl rather than a lactam. The third is the influence of the acyl side chain. Now what can we do to tackle this particular problem? How can we make acid resistant penicillin? Now we cannot do anything about the strain, that is ring strain, that is present in the penicillin, because we know having that strain bicyclic system is absolutely essential for its activity. So we cannot do anything about the first two factor, but we can do something about factor number 3, that is the influence of the acyl side chain. What we can do is basically, if we can place an electron withdrawing group on this position, means here if the R is an electron withdrawing group, what it can do, it can basically withdraw the electron density from this particular carbonyl. Basically what is happening here, the nitrogen lone pair of electron are coming and this carbonyl is attacking. So if we have an electron withdrawing group that is present here, so उस केस में क्या होगा कि these lone pair of electron, once they come back, this electron withdrawing group is withdrawing the entire electron density, as a result of which your penicillin will not undergo such sized products in the case of acidic condition. Okay, so let us suppose in the case of penicillin, what we have is a phenoxy group. Here the oxygen we know is electronegative in nature, right? As a result of which electronegative, it withdraws electron density from the chain, makes it much more stable to acidic condition. And then what we can do, at the alpha position we can put heteroatoms like amine group, chloro group. We can have this these type of group. We can have heterocyclic group. All these group, what they do, they withdraw the electron density from the carbonyl, as a result of which it makes penicillin acid resistant. Having amino group here will give rise to the class of amoxicillin, ampicillin, that those type of antibiotics. So how can we make penicillin acid resistant? Is by placing an electron withdrawing group next to the carbonyl. Okay, or by placing an heteroatom at alpha position, having an amino group, having a chloro group, or having a heterocyclic group. That is how we make them anti-acid resistant. Now what about the beta-lactamase resistance penicillin? Now we know that beta-lactamase, what it does is basically it hydrolyzes this particular bond. Now one of the initial approaches that was explored was, if they wanted to place a very bulky group here, what it can do, basically lactamase enzyme cannot approach this particular carbonyl because of the presence of the bulky group. But this particular approach has its own downside as well. Why? Because if this bulky group is very, very large, then this entire penicillin just cannot enter into the active site, active transpeptidase enzyme. That is the reason why there are a very, very limited number of beta-lactamase resistant penicillin, and here I have depicted few of them. So the first one is the methicillin. Okay, where what you have is this particular aryl group. So they are also regarded as a steric shield approach. Steric shield approach, it means that they are acting as a steric shield against the beta lactamase enzyme. And then what the other one is the neficillin, and the third one is the tamoxicillin. So here on the tamoxicillin, what you have is a carboxylic group. So this can also come under the category of the carboxypenicillin, which we are going to study later. Now here what I said, that on these this particular side you can have other groups. Here what we have is a methoxy side of the hydrogen, but you can see the stereochemistry between them. It is the cis, which is very, very important for it to act the way it does. Now apart from that, you can have these type of bulky groups as well. You can clearly see the bulk it can cause, but as I said, that such type of compounds has its own downside, that they are not very, very efficient. Okay, so as a result of which steric shield approach was not one of the most successful approaches. Okay, but you should know about that. Now we are going to talk about the broad spectrum penicillins. Okay, so broad spectrum penicillins are those penicillins which are active against both gram positive and gram negative bacteria. And if you want to know more about gram positive and gram negative, in my first video on when I discussed about the sulfur drugs, I discussed about that one. Okay, there are three classes of broad spectrum antibiotic. Okay, all of them have an alpha hydrophilic group. It means at this particular position here I am talking about, this is alpha to carbonyl. So what you have is an alpha hydrophilic group. Here what we have is an amine. Okay, what it does is basically it helps the penicillin to pass through the gram negative bacterial outer membrane. That is one of the important role this alpha hydrophilic group plays. Okay, to pierce through the porins of the gram negative outer membrane. So the first broad spectrum penicillin that we are going to talk about is the aminopenicillin. So this is usually the first line of defense. You must have seen, if you go to the doctor, they will either give you the ampicillin or they will give you the amoxicillin. The difference is on the alpha position, what you have is an amine and a phenyl ring, and here what we have is an amine and what we have is a phenol ring. So these are the two drugs that you will find, and these are acid resistance. Why? Because they have an electron withdrawing amino group. Nitrogen is electron withdrawing in nature. It is withdrawing the electron density, as a result of which these are also acid resistance antibiotic. However, they do not have any steric shield present. Okay, as a result of which both ampicillin and amoxicillin are sensitive to beta lactamase enzyme. So usually what the doctors do, they give amoxicillin along with the potassium clavonate, because the potassium clavonate takes care of the beta lactamase enzyme, and the amoxicillin can act as an efficient antibacterial drug. Okay, however, there is one of the problems that is with also present with the amoxicillin or ampicillin. And what is the problem here? It's the presence of these amino and acid group, and these two are ionized. As a result of which they are not absorbed greatly by the gut wall, because the gut wall is usually made up of the lipid, and if they are highly ionized, then that absorption is pretty poor. Okay, now one of the ways you can counter this problem is by somehow reducing the polarity of the compound, polarity of the drug. So what you do is basically you take advantage of the prodrug. What does the prodrug, what do we mean by prodrug? Prodrug means that actually the actual drug is released inside the body. Okay, so what you do is basically this acid is taken, is protected as a kind of an ester. So here you have a different variation of the R groups. So as soon as this particular drug is absorbed by the gut wall, it undergoes hydrolysis to produce the free carboxylic anion, which is absolutely necessary for it to act the way it does. Okay, so this is how you can take care of this particular gut wall adsorption problem, by taking advantage of the prodrug and by reducing the polarity of the one of the polar groups. One polar group we have is an amine, the other is the ester. Rather than protecting the amine, it is better to protect the acid, because this amine is what is making this particular entire drug as an acid resistant penicillin. And we know ester can be easily hydrolyzed inside the acidic environment of the body, and after the absorption of this particular drug inside the gut wall, it undergoes hydrolysis. Means these groups undergo hydrolysis to give you the free acid, so they act very, very efficiently. So the second broad spectrum antibiotic, or the penicillin, that we are going to talk about is the carboxypenicillin. So here, instead of the amine group, where what we have is an carboxylic acid, and we know carboxylic acids are also electron withdrawing in nature, as a result of which these are also acid resistant. Now in the first one, we can clearly see this is the carbinipenicillin, carficillin, and indyl carficillin. Now this these two, the second one, the carficillin and indyl carficillin, these are nothing, these are the prodrug of carbonicillin, sorry. Okay, and again this has like a hydrophilic carboxylic group, and this has a cis stereochemistry. Here the stereochemistry of the carboxylic group is very, very important. This is coming out of the plane, and it is only this drug which is efficient. Had this carboxylic group been going behind the plane, it would have been an inactive drug. So the stereochemistry of this carboxylic drug is absolutely important for it to act the way it does. Now, as I said that this carficillin and indyl carbonicillin, they are nothing but they are the prodrug of carbonicillin. Okay, so that it can be absorbed properly by the gut wall. And here what we have is an aryl ester. They did try the alkyl esters. So aryl esters means these are the aryl esters. Aryl esters were found to be much more susceptible to hydrolysis, because that's what you want. You want them to be hydrolyzed inside the body once they are done with their action. Okay, so they are found to have much more susceptible to hydrolysis because of the electron withdrawing effect of the aryl ring. Aryl ring का electron withdrawing effect की वजह से. Apart from that, you can also have the T-carcillin, where instead of the benzene ring, what we have is an heterocyclic thiopene ring which is present. Okay, so these are the various carboxypenicillin that are you can use. So here the last broad spectrum penicillin that we are going to talk about is the ureidopenicillin. So here, instead of the amine group, what we have is a urea moiety. So here on NR2, what we have is a urea moiety that is present here. So if we have this particular urea, it's called azolocylene. If we have this urea, it's called mesalocylene. If we have this moiety, it is called the piperacillin. So these are the one that are present, and mostly these have found to have much better properties than the carboxypenicillin. So these are the new breed of penicillin that are used in the pharmaceutical world that you should know about. Okay, so what they have instead of amine, so the first we studied was the amine at the alpha position, the second was the acid, and third one what we have is an urea that is present at the alpha position. All three of them you can see are electron withdrawing in nature, so which means they are acid resistant. Okay, now can we use penicillin in addition, in combination with other drugs? Yes. So one of them I said that the clavonic acid, which I clearly told why do we use that, because clavonic acid has a very high affinity for the beta-lactamase enzyme, so it inhibits the beta-lactamase enzyme. Now the second one has been found is this probenecid. Okay, this is nothing but a sulfur drug. Okay, right, this is nothing but a sulfur drug. So what it does when you take penicillin along with the probenecid, what it does is basically this drug competes with penicillin for the excretion mechanism. While excreting it, it competes with penicillin, which it also competes with penicillin for binding on the albumin. So what it means is that what it does, it basically keeps the penicillin inside the bloodstream for much longer time, as a result of which it's the antibacterial activity of the penicillin enhances, so which makes it a much better drug if it is used alone. Okay, so usually the penicillin at times is used along with the probenecid. Okay, so this is something that you should know about. So that's pretty much about the chemistry of the penicillin which I wanted to discuss. So in the next video I'm going to talk about the chemistry of the cephalosporins. Okay, so thanks a lot for watching the video. Have a very good day.